Hi everyone, I'm Victoria, and today I'll be talking about the creation of a deep learning framework, CROTON, which is capable of predicting CRISPR-Cas9 editing outcomes. CRISPR-Cas9 is a revolutionary gene editing technology that has broad applications in biology, biotechnology, and medicine. CRISPR-Cas9 works by inducing a double-stranded break, or DSB, shown here in DNA, which then leads to the activation of cellular repair pathways, namely non-homologous end joining, microhomology-mediated end joining, as well as homology-directed repair. So unlike these two other pathways, homology-directed repair involves a template sequence. This repair pathway is known to be error-free, and it can be utilized to implement precise genomic changes. And notably, more recently, it has been discovered that the editing outcomes of these two other pathways are also non-stochastic. It is known that their outcomes are determined by local DNA sequences, but the exact biological mechanisms underlying different editing outcomes is not yet well understood. So that is why we and other groups have turned to machine learning as a way of predicting template-free, so that's non-homology directed repair-based CRISPR-Cas9 editing outcomes. Before this work, there were 3 existing CRISPR-Cas9 editing outcome predictors called in Delphi, Forecast, and Sprout. However, all of these methods are reliant on existing and incomplete knowledge because they require both feature engineering and model engineering. So in this work, we proposed an alternative automated approach and trained our models on the data sets that these previous works generated and were based on. So we split the approximately 40,000 forecast data points in an 8:1 ratio to make our training, validation, and testing data sets. In addition, it was important to test our model CROTON on an additional endogenously generated dataset because forecast data was based on these Cas9 expressing artificial cells. It was based on these synthetic guide RNA CRISPR target constructs. So we also tested CROTON on the Sprout dataset, which was based on primary T cells, which is a cell line that has been widely utilized in therapeutic cell engineering. So in other words, the first objective of this work was to create an automated CRISPR-Cas9 editing outcome predictor, which we have named CROTON. So as you can see in this image, CROTON was trained on these raw one-hot encoded sequences representing the target sequences of CRISPR-Cas9 editing. CROTON processes a particular 60 base pair long target sequence, and then it outputs a list of these 6 editing outcomes, which are 1 base pair insertion and deletion probability, as well as deletion in these frame shift frequencies. And notably, since all of these editing outcomes were interrelated, we chose to utilize a multitask deep learning framework, which can outperform single task learning by leveraging features derived from multiple prediction tasks. So here's a closer look at how CROTON, which stands for CRISPR Outcomes Through Convolutional Neural Networks, was created using a highly automated machine learning pipeline. So to automate feature engineering, we utilize deep convolutional neural networks, or deep CNNs, which are capable of automatically extracting features from our input CRISPR-Cas9 target sequences. In addition to automate model engineering, we utilize neural architecture search, or NAS, a type of automated machine learning capable of generating other machine learning models. In particular, for NAS, we utilized a package named AMBER, which has been fine-tuned for application to biological sequencing-based machine learning. You can read more about AMBER in this work at the bottom right. So the first thing that we did is we analyzed whether NAS could design deep CNN architectures effectively. So we compared NAS engineered CROTON, which is shown in red in this figure, to 50 other randomly selected architectures from the model search space, which is shown in gray in this figure. So here you can see NAS generated CROTON achieved higher Pearson's correlations. In other words, it outperformed the other randomly sampled CNNs. So NAS designed CROTON, in fact, achieved a Pearson's correlation of greater than 60 for all prediction tasks, and that included a Pearson's correlation of 88 for deletion frequency and 91 for 1 base pair insertion probability prediction. So this overall indicates that NAS was an effective designer of multitask deep CNN architectures. And here on the right are the layer selection probabilities for different deep CNN layers as chosen by NAS. So you can see that convolutional layers, so these conv layers, were consistently favored over pooling layers, so max and average pooling, as well as the identity mapping layer. And after layer 2, this dconv8 layer, shown in green, or the dilated convolutional layer with a kernel size of 8, was consistently favored. So together, this indicates that precise feature locations were conserved in our model and were important to CRISPR-Cas9 editing outcome prediction. We also assessed the accuracy of CROTON by evaluating whether it made accurate predictions above or below the median value in each task dataset. So here you can see AUC-ROC curves for CROTON's predictions. On the left is CROTON's performance on the test forecast data, and on the right is CROTON's performance on the held out test sprout T-cell dataset. So here you can see by the AUC values listed here and here that CROTON's accuracy was conserved across cell lines, and in fact, CROTON achieved AUCs of greater than 0.75 for all prediction tasks, and that included an AUC value of greater than 0.9 for deletion frequency and 1 base pair insertion probability prediction. We also compared CROTON to other expert design models on their predictions for the Sprout T-cell dataset, which CROTON was unexposed to during training. So shown in these tables are values describing the correlation between each of these 4 models' predictions and the actual Sprout data points. So it either shows the Pearson's correlation or the Kendall's tau value. So overall, you can see that CROTON substantially outperformed all models on most tasks, and notably CROTON performed on par or even outperformed Sprout, which was itself trained on the Sprout D-cell dataset, which was held out for CROTON. So CROTON is publicly available at this GitHub link. So this repository contains some of our model creation scripts, as well as code and instructions to set up this Django web framework. So essentially here, a user simply inputs their sequence, they can press predict, and then you can see the different editing outcomes are outputted here. And since CROTON was performing so well and does not utilize any manual feature engineering, it was important to elucidate how CROTON made predictions from a raw input sequence. So we conducted a technique called in silico saturated mutagenesis for CROTON. So shown here are some representative plots for 1 base pair insertion and deletion probability prediction, as well as deletion frequency and frameshift frequency. So essentially in here, these plots show the average importance values that CROTON computed over multiple sequences in the test dataset. So importance values are assigned to every nucleotide on the input sequence, and essentially the larger the letter, the more important the nucleotide position. So for instance, this nucleotide is really important because these letters are very large. So consistent with prior reports, we found that the positions of nucleotides relative to the PAM site, which is around position 33 in these figures, were important to CRISPR-Cas9 editing outcomes. So in particular, base pairs or nucleotides upstream of the PAM sequence were important, or generally the most important to CRISPR-Cas9 editing outcomes. And more specifically, our analyses support previous studies that have shown the nucleotide 4 base pairs upstream of the PAM, so this one, this one, this one, and this one, were the most important, had the greatest effect on CRISPR-Cas9 double-stranded break repair. And this brings us to our second research objective, which was making CROTON variant aware. We work to utilize CROTON to explore how genomic variation affects CRISPR-Cas9 editing outcomes in a clinical context, essentially in a patient context. So this analysis is especially pertinent because CRISPR is currently being used to inactivate genes in several clinical trials. And this is often through cell therapy, where researchers take cells from a patient, they inactivate a particular target gene, and then they reinsert these cells into the patient. So we investigated 4 genes. 2 have applications in antiviral therapy, and those are ACE2 and CCR5. ACE2 is a receptor for the SARS-CoV-2 virus, and CCR5 is a receptor for HIV. And both of these genes have been considered therapeutic targets for these viral infections. And the other two genes were PDCD1 and CTLA4, which are important in cancer immunotherapy, more specifically in immune checkpoint blockade. And notably, the first CRISPR-Cas9 based clinical trial to clear safety concerns in humans was just reported last year, in which researchers inactivated PDCD1 in T cells. So in that study, Xue et al. Essentially utilized CRISPR-Cas9 with the goal of treating late-stage non-small cell lung cancer patients, and their trial was deemed safe and feasible. So the specific subset of genomic variation that we chose to focus on were single nucleotide variants, or SNVs. So there are around 10 to 15 million common human SNVs, which have the potential to impact CRISPR-Cas9 editing outcomes. So for instance, for some reference genome, there may be around a 90% one base pair insertion probability, and the alternate genome, which contains an SNV, may only have around a 40% one base pair insertion probability. So these are essentially differing CRISPR-Cas9 editing outcomes, which then may lead to differing gene inactivation efficacy, which then may lead to differing CRISPR-Cas9 based cell therapy efficacy, which are already being implemented in the clinic. And notably, SNVs may be closely connected to individuals of a particular racial or ethnic background. So our investigation may be a critical step in making CRISPR-Cas9 based cell therapies not only more effective, but also more equitable. And since CROTON is based on CNNs, it is sensitive to changes in target sequences at a base pair resolution. So we utilized CROTON to evaluate the effect of SNVs on one base pair insertion probability prediction, and we found a significant impact. So here you can see a table describing SNVs with a particularly high impact on CROTON's one base pair insertion probability predictions for our genes of interest. So you can see the absolute difference between CROTON's predictions for the reference and alternate genomes for the reference and alternate sequences, which differ by an SNV, are all greater than 0.36. We also further analyzed CROTON's one base pair insertion probability predictions for PDCD1, because this is the gene that has already been targeted in that clinical trial that I previously mentioned. So here on the x-axis are the 211 different PAM sites on PDCD1, which are colored by exon. And on the y-axis is CROTON's predictions for one base pair insertion probability. So the black horizontal line in each of these clusters indicates CROTON's predictions for the reference genome, and all of the circles indicate CROTON's predictions for various alternate genomes. In addition, you can also see these two orange boxes, which indicate the targets of the two guide RNAs that were utilized in that non-small cell lung cancer clinical trial that I previously mentioned. So you can see that looking overall, zooming out at the whole gene body, you can see the one base pair insertion probability in PDCD1 varies considerably across the coding regions. In addition, for this guide RNA utilized in that clinical trial, there is a single nucleotide variant that seems to have a substantial effect on its editing outcomes. So future studies may reveal whether there are better guide RNA pairs than those utilized in that clinical trial that would be more effective for CRISPR-Cas9 editing and more consistent over a patient population. So in conclusion, we have successfully created a CRISPR-Cas9 editing outcome predictor that is automated and also variant aware. CROTON is automated, publicly available, and outperforms existing methods, namely in Delphi, Forecast, and Sprout. In addition, the successful creation of CROTON provides evidence for the effectiveness of synthetic construct-based data generation, which is often really helpful in generating the large amount of data needed for deep learning in a biological context. Our study also provides evidence for the potential and usefulness of neural architecture search or other automated machine learning in genomics. In addition, we found that single nucleotide variants significantly affect genome editing outcomes, making our model variant aware. So CROTON's predictions indicate that SNVs may be important factors to consider in CRISPR-Cas9 genome editing, especially in clinical trials that may have patients that harbor these variants. And to our knowledge, this is the first study that considers how naturally occurring variation may affect CRISPR-Cas9 gene editing outcomes. And a CRISPR editing outcome predictor that is sensitive to genetic alterations at a base pair resolution like CROTON could be more and more critical in the future to make sure that these gene therapies are effective for everyone and may even be tailored to individual patients. And finally, ending with my acknowledgements, a really big thank you to my mentor, Dr. Frank Zhang, who has been invaluable in my work on this project, as well as Professor Olga Trayanskaya. Also, thank you to all the other members of the Trayanskaya lab for their helpful discussions. Also, thank you to all of you for listening, and I'd be really happy to take any questions.